профессор Университета Сан-Паулу Александре Ferreira.
Professor of São Paulo University, Alexandre Ferreira Ramos, characterizing the regulatory logic of transcriptional control at the DNA sequence level by ensembles of thermodynamic models.
Hello, good morning. So it's a great pleasure to be here. I'm honored to come. And thank you for the organizers for making my travel to Moscow so smooth. So today I'm going to talk about framework that we developed for characterizing the regulatory logic of control of gene transcription by means of an ensemble of DNA sequence thermodynamic models. This model was, they were validated using Drosophila embryos. And one may ask, what is its relationship with the artificial intelligence journey here? So sometimes to see, one needs to go beyond the Earth to see that it is blue. So it's perhaps we could look at biological systems and see that to each element involved in a typical artificial intelligence process, there will be some correspondent in biology, and that correspondent can be useful for enhancing our artificial intelligence models. For example, by offering inspiration on finding loss functions, or to figure out good dynamical systems that would enhance optimization processes. So the outline of this talk is, we first would like to introduce some concepts which can be useful for us to state the cells as entities, computing entities, specialized in learning in the statistical sense of the term. Then I would like to demonstrate that, to show that, discuss that by means of some experimentally established facts. Then show our technique for understanding the regulatory logic of transcription in metazoans, multicellular organisms, and in the end, to present some concluding remarks. So, in short, if we were considering computing as Turing devised, we could consider that we need an alphabet, we need an infinite tape, we would need a states machine, and we would need a head that would be capable of reading and writing into this infinite tape. And our state machine would react to the information contained into that infinite tape. If we look close in the flow of genetic information that is taking place inside the cell, we can map each of the elements that are happening there as a Turing machine. So we would have the tapes that can be either the DNA or the RNA. We have an alphabet, which is composed by the nucleotides, A, T, C, G. We do have a writing machine, a read and writing machine, which is the polymerase, which takes the information that is in the RNA and translates it into a sequence of amino acids. So the sequence of amino acids at some moment is going to become a protein into its folded shape, which will enable it to perform its function inside the cell. And we do have a states machine, which can be written in terms of the genetic code, which is setting the translation of a given triplet of codon of nucleotides into a given amino acid. So with this in hand, this is a very explicit formulation of molecular biological process in terms of as a computation process. However, There is one extra thing in the biology, which is that the information, it is regulated, so it doesn't come up anytime. It can come up sometimes or not, depending on the context. And that was seen back in the 60s by Jacob and Monod, who were investigating this little system of gene expression called lac operon, and in which they were showing that if there is presence of lactose, so this system is going to be expressing. However, if there is no presence of lactose, that this system will not be expressing. So regulation here happens by physical blocking. So a repressor protein binds into a specific DNA site, and then it avoids the information to be expressed. So it's a genetic switch. So with those two examples of computation in molecular biology, we can have a phenomenological understanding of how we can consider what is going into inside the cells as a computing process. And that perspective was actually formalized by Lieberman from here in Moscow, and who did not only the theoretical but also several experiments to show that the cells are behaving as if they are following some algorithm. And this algorithm then must be discovered. So if the cells are operating algorithms physically, how were those algorithms learned? So when we are talking about learning, and since we are talking about cells being computing systems, what we should be, the perspective of learning that we should have is learning in terms of a computer program that improves its performance with experience. So this is, or in terms of statistical learning theory, the cells need to discover some function, biochemically, some function that enable them to perform well in the environment where they are located. So how was that learning learned then? Well, we can consider evolution. And two problems. So the improvement of performance happens as an adaptation. And of course, there are multiple ways of learning how to handle the environmental conditions. So there's also speciation. So besides learning how to adapt to a given environmental condition, it is possible to adapt in several different manners to that. That is interesting because if we are looking for ways of defining risk functions or loss functions, adaptation is one way of doing that, and hopefully we will be able to use that in our favor for improving artificial intelligence. Besides, we will see that there are multiple ways of doing this. So if the cells are learning machines, we should discover the biological laws of what are the error functions, what are the equivalents of statistical models in terms of the internal machinery. So look, we are always talking here about some mechanisms that is implementing a given functional, computing a given functional. So in other words, we are talking about somehow Church-Turing hypothesis, physical realization of it. What are the input data, the ever-changing environmental conditions? So we are always learning. We are always living things are always improving. What is the optimization process? So this is interesting because is there any sort of a special dynamical systems that is defining trajectories within the, let's say, parameter space of DNA that are making learning fast enough to avoid that the living thing will be extinguished because it must learn fast enough. So let's go back to the information flow and remember that a genome may have billions of base pairs. It encodes multiple enzymes and it also has numerous biological mechanisms. And this part here is important because once a DNA learns how to handle environmental conditions, it does it encoding several functions. Let's consider in terms of representation theorem. So it's being done, it's adaptable, as we saw in terms of that example of lactooperon. If we inject lactose, it will express, otherwise it won't. However, there is a challenge here. If we look at the genetic code and we look at the DNA, we know exactly what are the proteins that are being encoded. However, if we are taking a look to understand how cis regulatory code works, that means how our system, the cells adapt to the differential information that are coming in, then there is no clear picture of it. You know, I cannot put you guys a table like that states machine back on the previous slides. So there is a challenge. It's an open question that we need to solve. And in order to solve it, it is useful to have a good model system, like this beautiful fly here called fruit fly, Drosophila melanogaster, which life cycle is about 10 days. There is an interesting property of it, because when it is in this phase here as an embryo, when the cells still do not have any membranes, they do form some patterns of expression of several proteins along its anterior-posterior axis, which will correspond to the formation of segments which will encode the organs that are going to be part of the adult fly. If we look a little bit closer into this embryo here, what we see is that after a series of cell divisions, nuclei divisions, all the nuclei will be in the internal surface of the embryo, and there everything is going to happen, which means all the pattern formation of proteins that will give rise to the segments into the adult will appear. So this is a molecular resolution perspective. This is nuclear resolution perspective on the expression of several proteins inside the embryo of the Drosophila. And this is a maternal protein, which will regulate the expression of those genes here that will form gaps. So here we do have a gradient. Here we will have a gap pattern of expression. That's why those genes are gaps. In the adult, there will be a missing part like a gap in the adult. And then there will be the so-called pair rule genes. They will form stripes like that, 7 stripes. And in the end, there will be the segment polarity. All of this regulation happens hierarchically, so the proteins that are above, they regulate the downstream and so on and so forth. So, and we will focus on the expression of this particular gene, Eve, and we will use it as our model system to try to understand how biological systems are learning how to encode a multiplicity of functions to deal with the environment within which they are. So if there is an information, as I said, information is not only encoded in terms of the sequence for a given protein, but it is in a given gene loci. It has a lot of DNA sequence that are not responsible for encoding for a protein, but actually to encode for regulation of the expression encoded here, of the information, of the expression of the information encoded here. So here we do have, this is the encoding, this is the regulation, including regulation of this protein, not only during the embryo phase of the life of this fly, but also when it is aged during its late time. That are also in the heart and so on and so forth. So there are specific, each of those segments are encoding regulation of this information to be expressed in specific contexts, and each of those green ones are encoding for the regulation of expression of those stripes here. And if we look inside each of these squares, what we will see is a multiplicity of binding sites for different transcription factors or proteins that are regulating the expression of this gene and ensuring that it will appear with this pattern at the proper time, in the proper quantities, and at the proper regions of the embryo as well, so that it develops viably. It's good to know that since we are talking about a model that we want to validate experimentally and with which we would like to learn on the mechanisms of life to evolve and cope with its environment, data is obtained by immunofluorescence, which will emphasize the presence of specific proteins or its absence. This data is collected by microscopy and quantified, and then we can get a pattern, a spatial pattern of expression of a given gene along space and time by looking either at transcription factors, that is proteins, or mRNAs. And now another thing is to understand the regulatory mechanisms underpinning this. So what we have is multiplicity. The expression of the information not only depends on the binding of the protein to the DNA as it happened for the lac operon in the bacteria, but we are talking about a more complex organism. So it has a lot of additional effects of the interaction among bound proteins that are driving by increasing or reducing the expression of this gene in differential contexts. And this is important because that is what is providing the Drosophila embryos to learn how to express information into a much more complex manner than it would be if it was just a bacteria. So this is just a pictorial representation of the mechanisms of regulation, and what is important to emphasize is that there is not only the binding of the regulatory factors to the DNA, but also their interaction once they are bound. And then this is a view of what this modeling is doing. I regret that I didn't put the equations, but if we were looking at the, so for a given input, so a given regulatory profile in a given position of the embryo, this is along the anterior-posterior posterior region of the embryo. If we are looking at a given position, it will have a specific regulatory profile as provided by the concentration of the transcription factors. This is two different positions of the embryo. The DNA is the same, and if we were looking only at the affinity between the transcription factors and a given DNA region, some regions would be more favorable for the binding of a given protein than others. However, because of the concentration of those proteins, the vertical bars here indicate how activated a given region of the DNA is to induce transcription. What we would see is that there is a convolution, and this convolution will modify a preliminary configuration into refining and finally indicating the region that is going to be activated and inducing expression of this gene. So this is some sort of, we could say that this is some sort of, we could consider a representation theorem and use this as a neural network, of course. However, an interesting thing is that differential regulatory profile would cause adaptative weights for the neural network. As you can see, we can we can recover the pattern of expression, but we can also indicate what are the regions of the DNA that are inducing the expression of each of the peaks of the EVE gene. And this is really important because now we can sort of interpret how this function is leading to a observed pattern of expression. And we can also consider this problem as an optimization. So we do an optimization in order to recover that pattern, which has two steps: a regression that recovers the pattern of expression, and a classification that aims to recover the regions that are inducing the expression and classify them as corresponding or not to experimentally established facts. So some of the optimizations will be kept, others will be discarded because this pattern here is caused by the activation of this region of the DNA, and that does not correspond to experiments. All right, so, and if we look at the parameter space, we see that there is a wide range of parameter values that enable the same patterning. And that leads us to the second question. Let's suppose that this is not an artifact, but this is an actual biological phenomena, and this is some sort of a prediction. So is it possible that a differential regulatory profile would provide the same patterning? And if we do this by looking at different species and taking the DNA of a different Drosophila species into Drosophila melanogaster, so we are taking the DNA of another Drosophila, another embryo, Drosophila jacoba, Drosophila erecta, Drosophila obscura, and then we are trying to see if we take this piece of DNA that regulates the expression of the Eve gene at the region that forms the second of those 7 stripes. Would it be possible for us to recover, changing the DNA, recover the same pattern of expression? And what we see experimentally is that, yes, it's possible. So you change the regulatory region, that means you are changing the weights of your neural network, and you are still getting the same output of your function either way. So that's a good thing. That means that eventually finding different regulatory regions causing the same pattern of expression may have a biological meaning, and then that leads us towards the need of using ensembles of models for multi-parametric systems. But if, let's say that we decide to go beyond, and instead of looking in Drosophila embryos, we decide to look in Sepsid, which is a very, very different species, quite a way separated from Drosophila. If we look at their DNA, the loci for even skipped gene, they differ a lot. And then let's look only at the stripe 2. You see, this is in Drosophila, this is in Sepsid, quite different. And then, if we set the DNA region of the Sepsid into the regulatory profile of Drosophila melanogaster, put this piece of DNA here, and we look to see if we get the stripe 2, look, we do get the stripe 2. So what it means is that we can have a degeneracy in terms of the regulatory region and still get the same functional, get the same information, the same genetic information, or the same function into our system. So that is actually interesting because if we look at the DNA sequence differences, we see that Sepsid is quite away from Drosophilids. So it's remarkable that changing DNAs for so differently, we still can get the same pattern of expression. And that can be done using several different species, using the same model with the same parameters, just keeping the same regulatory profile, that is the same concentrations of transcription factors driving expression of the gene, but changing the DNA sequence of the regulatory region. So what we can do is to get the same very useful patterns of expression, and we can recover what has been observed experimentally, either using a Drosophila species or Sepsid species. So that being said, I would like to come back to the understanding that, so can we use ensembles of models, ensembles of optimizations to understand regulatory logics, and can we learn a little bit more about how living organisms, how cells, they learned to process environmental information to improve their performance in that environment, and how they do navigate through their parametric space so that they can get good optimizations within a sufficiently short time so that they will not be extinguished? The answer is yes, and if we were looking for risk functions, what would be the ones? Well, we should search for invariant quantities. Interestingly, is that if we are talking about a Drosophila, we are talking about some thousands of genes. Let's say that those would be, and then we would get several gene networks, and those networks could be mapped as neural networks. So we would be talking about several neural networks, each of them having one invariant quantity. You know, the pattern formation is only one of the invariant systems inside the Drosophila embryo. And talking about gene networks, this brings us to the equivalent of the statistical models for predicting. You know, a cell must cope with its environment, so it reads input from environment, rewire it fastly, its internal machinery fastly, and adapt to it, respond to it. One example is if we had some milk during the morning, we promptly must know how to metabolize that, and that is done by activating lac operon. And then two interesting questions would be, how is the optimization process performed? So how do the cells fastly enough learn how to handle new stress? And that is a nice challenge for mathematics. And let's say, is there any sort of special dynamical systems? Who knows? So this is a nice question to be responded, and I'm pretty sure they will have some impact for the artificial intelligence field. So with that, I would like to finish and thank Alan Sabino, who did the work on the characterization, and Rieli, who helped on the optimization using simulated annealing, and Professor John Reinitz, with whom I have been collaborating to this project for a long time. So thank you so much for your attention.
Alexandre Ferreira Ramos.
Alexandre Ferreira Ramos, thank you.